Mr. Gill's revision egg.
Okay, so lesson number 6. We're going to carry on with the cooked egg white. The cooked egg white here, as you remember from the last one, is basically just egg white albumin protein that's been taken beyond its denaturation point and has gone solid. Right? In other words, it's cooked. But it's still protein. And one thing we can do with proteins is we can break them down using enzymes, protease enzymes. And you are expected to know how protease enzymes take long chain polypeptides and break them down into small chain amino acids. Small chains of amino acids. So what I've got here is actually a very powerful protease. That's where my pineapples come in here. Pineapples contain a very potent protease enzyme called bromelain. Okay, now bromelain is a protease that is a endopeptidase, so it cuts within the chain. You are expected to know the difference between endo and exopeptidases. It chunks those large polypeptides up into smaller bits. So here's some I made earlier. So this is just some blended jarred pineapple. And I'm just going to see if I can suck a bit of this juice up here. Like so. Pop it in with our egg white. Now, this won't be as effective as fresh pineapple because, of course, the pineapple before it's been in the jar, been in the can, has been cooked. It's been heat treated for sterility, and it's been canned. So I wasn't sure that this was going to actually do anything. But there does appear to be enough bromelain activity in this pineapple juice to break down this protein. And to illustrate that, I can show you what I did yesterday. By the miracle of Blue Peter style. So, here's our two. In the one hand here, I've got yesterday's and this is today's. So as you can see, this one, solid white egg with the pineapple juice on top, nothing there. This one here, and I have taken some pictures along the way to show you, has actually broken down quite nicely. So the bromelain, what little is left in there, has actually attacked the egg yolk, egg white, and broken that down into shorter chains and start to digest it. And if I left that long enough, I'm sure it would digest all of the white in that tube. Okay, so of course you are expected to know how that works. What we've got, bromelain's got a very broad, accommodating, active site that takes in lots and lots and lots of amino acid chains. You form an enzyme substrate complex. Right? You then get the lysis of the peptide bond, the addition of water. Right? It's a hydrolysis reaction. Now I did try a different experiment to show you as well, which is more like one you might get on an exam, which is to make a little plate of cooked egg white and cut holes in it. And then put the pineapple juice in there to experiment with concentration. Because remember, the variables you're expected to know about are temperature, pH, how they affect enzymes, of course, but then concentration of substrate, concentration of enzyme, and of course, inhibition. Now, I don't think there's anything fancy enough in the kitchen to be an enzyme inhibitor specifically for bromelain, but what I did do was experiment on this, and I'll show you some photographs of this, with cutting some little holes in the egg white, and adding the pineapple juice in to see if it would digest away at different concentrations. And what it actually has done is it has done that. It doesn't look overly convincing because the egg white has the egg white has dried out and started to crack. But you can see those holes right in there where the bromelain has had some impact. So not the most effective experiment I've ever tried to do at home, but it illustrates the point. And that's what we're talking about in this lesson. That is enzyme action. So you're expected to know once again about inhibitors, enzyme concentration, substrate concentration, temperature, and pH. And that is lesson 6.
Mr. Gill's revision egg.
P.S. This is what happened to the egg a day later. Just showing how strong that bromelain actually is. As you can see there, completely broken it down. Hooray for pineapples.